From Microbe TV, this is Immune, episode number 37, recorded on November 12th, 2020. I'm Vincent Raccaniello, and you're listening to the podcast about the body's defenders against disease. Joining me today from Ithaca, New York, Cindy Leifer.
Hello again.
You're in front of the window again. Good job.
I am. I'm back home again.
From Durham, North Carolina, Steph Langle.
Hey there. Good to be here. I'm in my office today, not at home, and excited for some great content we have.
And from Madison, New Jersey, Brianne Barker.
Hi, it's great to be here. I'm also at home in my little podcast area, and I'm excited about the content too.
We have 5 guests today who are going to help us to celebrate and explain the upcoming Black in Immuno Week. And let me introduce them for you. From the University of Texas, MD Anderson Cancer Center, Alexis Mobley. Welcome to Immune.
I'm glad to be here. Thank you.
From the University of California, San Francisco, Joel Babdur. Welcome.
Thank you so much for having me. I'm very excited to be here.
From the University of Chicago, Elaine Kouame. Welcome to Immune.
Hello. Happy to be here. Thank you for having us.
And from my old stomping grounds, the Icahn School of Medicine at Mount Sinai in New York City, Justine Noel. Welcome to Immune.
For that introduction, I'm excited to be here.
When I went there, it wasn't Icahn, though. He hadn't funded it. It was just Mount Sinai School of Medicine. But it's more or less the same place. And from the University of Pennsylvania in Philadelphia, Yvonne Jean, welcome to Immune.
Yes, thanks for having us.
And so we have people all over the country, and that's the power of the internet. I don't know what we would do during this pandemic if we weren't able to connect this way. But then again, there weren't podcasts before the internet, so there you go. So, Steph, I'm going to hand it over to you and tell us what's happening here.
Yeah, so this is really, I was looking at Twitter one day, and I think it was Joelle who was the first one who came on my radar to tweet about Black in Immuno Week. And this really comes on the heels of a lot of advocacy work that has been done really at a grassroots level for raising up really excellent researchers, Black researchers in particular, in STEM fields. So I don't know if I can remember the first Black in X week. I don't know. Joelle, what was the first one that came on your radar?
I'm not sure that was the first one, but the first I saw was Black Birders Week.
That's right. I would say that was for me too. And I was able to really kind of follow people that I had never followed before, which really was the point. And it's an excellent representation of what's going on that I believe universities have not been able to raise up to the task. Even PIs, staff, faculty maybe haven't been able to do what the people on the ground of Black and X Week have been doing to really raise awareness about Black scientists. And I think that this upcoming one is going to be my favorite because it's going to be Black in Immunology, Black in Immuno Week. And that's going to be held November 22nd to the 28th. We're really looking forward to highlighting 5 researchers. They're going to talk about their advocacy and their science because both of those domains live within them and really should live within all of us. I think that it is an important topic, particularly on the heels of the death of George Floyd, and kind of a reckoning within academia, of course, comes too late. And even now, when I think about, and you all can tell me what you think about this, but I think it's dying down because things happen and we have these universities want to grab a hold of the moment, and we have, meetings, and we do different things, but then it kind of goes away. And I think I'm really liking these efforts to kind of keep things in the spotlight, but it can't just be Black scientists with the burden on their shoulders to maintain advocacy, because at the end of the day, We started this mess. We need to help fix it as ally in academia. So I think, coming up as somebody in science who came from a privileged background, like it's incumbent upon me to do something. So I think, we'll do a little round robin with the guest to talk about maybe what diversity means for you and removing racism in science, because at the end of the day, we're people, we come from different backgrounds. Science is not political in the sense that a piece of data is not political, but in the context of science that we do as people is political. We can't not talk about it. And I think there's science to back that up. The Black and Immuno team had curated a really excellent list of different research articles, commentaries that kind of show us in the data why, how Black scientists get excluded even from the start of, undergrad. I think it probably goes primary school as well. So we'll share some of those resources. And I don't want to hog the spotlight. We can come back around because I have some other thoughts, but I don't know, Cindy, Brand, Vincent, if you have anything.
Sure.
Yeah. Go ahead.
So I'm really excited about Black and Immuno Week and all of the efforts of Black in insert and something here weeks, because I think that it's great for us as members of the Academy to think about all the ways that universities are not necessarily level playing fields for people. And, there are so many different things that are getting in the way of members of lots of different groups and so many different challenges that those groups are facing. And whether that comes in the case of those big challenges or whether that even comes in terms of, microaggressions. I don't know if the opposite of a microaggression is a macroaggression or how we call that. But in any case, we have a lot of different things that are standing in people's way that are not allowing them to be successful scientists. We as immunologists spend an awful lot of time talking about the importance of immune diversity and the diversity of immune cells and making a response. And just like it's really important to think about that, immune diversity, as we think about all of the problems that we as scientists want to solve, we need to make sure that we're including everyone, everyone's skills, everyone's ideas, to make sure that we have the best possible ability to actually get this work done. If we're sort of shutting out or putting up barriers in the way of certain people, that's not going to help us to actually get our science done because we aren't going to have everybody involved. I know that I've been spending a lot of time thinking about ways that I can work with undergraduate students and reading a lot of literature. I was super excited when I saw some of the literature that you all shared about different types of teaching that can be innovative and that can really help other different groups of students to help underrepresented students do well. And I also think that I'm really excited to go on Twitter each week during Black and Immuno and some of the previous weeks and find new people to follow and also find new people to start talking about with my students. And making sure that, my students are really understanding all of the people they could be like in the future, which is, I've tried to do before, but this has given me so many more opportunities. And I think things are going to get so much better. So I'm really thankful for all of these efforts.
Yeah, so I would add to that I think that you made an outstanding comparison using the immune diversity to look at diversity, because that's like the best possible analogy being it, Black and Immuno. I think that's fantastic. And the whole idea that the immune system doesn't work unless all the parts are working, right? And they all complement each other and they all work together. And so I think that's really important. I think from my perspective, it's really interesting because I'm at Cornell University, and I don't know if you know, probably not. The founding motto basically of Cornell University is any person, any study. And so from the foundation of this university, there has been a drive to make sure that everyone has an equal opportunity. And even then we struggle, right? And so I'm at an institution where that's the foundation of the institution. And there are so many programs to promote faculty diversity, to promote student diversity. And I actually have a grant from NIH to promote faculty diversity. And so we have a number of initiatives, and we can talk about that later. But so there are many, many ways in which we have all of these programs and these opportunities, and yet we still struggle because the number of individuals who are not white who are on our faculty is still slim, right? It's still low, despite the efforts. And I think what we can take away from that is that this is not a passive process. We can't just wait for things to happen. We have to have actual practices and policies in place to break down those barriers that you were talking about, Brianne, to actively recruit and actively promote individuals of diverse backgrounds. And it's, we're here talking about Black and Immuno Week, but it's all diversity, right? Military disabilities, sexual orientation, race, religion, all of that together impacts everything we do. And so having all those different diverse voices is really important. And I think, actually, I know that it works because the studies have shown that when you have diverse teams, they're more successful than teams that are like each other. So it's when people have different opinions and different backgrounds and they challenge the way you think and get you to broaden your perspective and broaden the types of solutions you might have to a problem when it just elevates everyone.
I think the best thing I can say is to repeat a letter that we got on Twiv a couple of weeks ago. We had Kazmikia Corbett on, and this person wrote that they are from a country where they're brought up to be racists, and they make no bones about it. They don't hide it. And this person said, listening to Kazmikia, he felt his racism melting away because he realized what this person could do and overcome all the things he'd been taught when he was growing up, right? So the most important thing we can do is to showcase what Black people can do, not just to show what they can do, but to show young Black people that they can do the same thing as well. And, we have a lot of listeners, so I'm happy to have our time on these podcasts devoted to that.
Yeah. And the one thing I just want to point out with all of that is I am so excited to allow for a lot of opportunities like that. And it also sort of makes me really sad that those things have to happen in the first place. And also that anyone has to feel like they need to be some sort of representative, or that anyone needs to sort of disprove any of those types of ideas. That makes me really sad. And I'm honestly excited that I get to hear about 5 new people's science today. Yeah. And I get to hear about 5 new cool projects that are going to really help me, learn more and think more about immunology. So, let's not think about this as just about, we're talking about Black and Immuno Week. We're also talking about super cool immunology.
Oh yeah.
Absolutely.
And and one of the things I would add to that is when you were talking, it reminded me of this thing. And I'm going to butcher this, and everybody will come back and yell at me this, but there's something about Ruth Bader Ginsburg, the late Ruth Bader Ginsburg, made a comment about, like when will things be equal? And she said something to the fact of, when there are 7 women on the Supreme Court or 9 women on the Supreme Court, right? And the idea was that, it was not a surprise when it was all men, right? It was a norm. And what we want is to get to the point where it doesn't matter who's on the panel, nobody notices, right? That everybody really truly is equal. And, so we get to the point where if we have, 5 phenomenal keynote speakers who are all diverse, you don't notice that there's no white man there, or you don't notice the race or the gender of people. You just notice the science and that it's amazing, right?
I would say let's get 9 Black women on the Supreme Court. I think I'd like to see that.
Right.
Well, that's great. I mean, I think, and to even as co-hosts of Immune, I think, we recognize too that within ourselves that we have things that we have to work on internally, and it's an active process. And I'm excited to hear, the advocacy and the science of these 5 great individuals here. So I think we'll pass it to them, and you guys have a lot you want to talk about, and we'll interact and ask questions because I think that's going to be a good discussion.
Yeah, so I guess I'll start with my science. So is that where we want to go first?
Whatever.
I think we wanted to know what immune cell you were.
Oh, yeah. So the immune cell that I am. Sorry, it's been a while since I looked at the list. Slept since then a little bit, but not a lot. I would definitely say that I am an innate lymphoid cell, particularly group 2. Very convenient because I also researched that cell. So I like to think of them as rare but mighty, small and tiny. I don't know if anybody watched the entire Cells at Work. But they introduced the NK cell and she's just like this great battalion woman that just really, she's smart, but like she doesn't deal with any mess. And I actually loved her introduction. So once it's safe to go out again, I do plan on like cosplaying her at some point because I thought it was the best. So, I mean, I feel like all the ILCs are kind of in that family where, they kind of say, okay, you can calm down, T cells, you can have your moment, but like we're actually the stars and make it a lot easier for you on the tail end. And so that's definitely how I like to view myself and why I love the ILC2 so much.
Yeah, those ILCs. And I mean, when were they discovered? Relatively recently.
Yes, within the last 10 years, actually. 2010 was when they first started hitting the scene. So
wow.
That's like a neonatal cell in immunology.
Yeah, pretty much. So it's really fun because I like it because for my science, I don't have to be married to my hypotheses because there's not a lot out there. So whatever happens, I'm like, it's a new discovery. We're having a fun time, as long as like the data starts trending, I mean, we're good. So, yeah, I'm really excited about it. It's a cool place to be.
How did you find immunology and start getting into this field?
So for me, for immunology, it was an accident. Actually, my best friend, she was going to pharmacy school and she needed immunology as a prereq. And we took all of our classes together. And it was actually one of the hardest courses on our campus. And I told her, I'm not doing immunology. And I was like, if I fail the first test, I'm out. Like, you will be here by yourself because you're not messing with my GPA. And surprisingly, as soon as I got into it, I fell in love. And just the complexity of it and how beautiful it is and how, we discussed before, everything has to work in concert. And if one thing is out of balance, then, I mean, it throws everything out of balance. But I could also see myself in immunology just with personal health issues that I'd been going through. So it was cool that I'm like, oh, this is what's going on. And so from there, I just ran with it. I had to, because I needed to know more. I had to do more. So.
That's so great.
Yeah, that's awesome. And I mean, so after you really fell in love with immunology, what was kind of your journey since then?
So since then, after undergrad, I applied for PhD programs and everybody said no. So with another kind of happenstance, there was a new program in biotechnology at Texas Tech for a master's degree. And my advisor there, I would say, she was in touch with the new director and she's like, this would be a great program for you. And she's like, they do cancer immunology, which is kind of what you're wanting to get into anyway. And it's a new program and they're looking for students and like, you would be phenomenal. So I got in contact with them and got in. So I ended up getting my master's degree in biotechnology in Abilene. I got an introduction to biotech startups, industry work, more of the bedside type things, patent law, FDA approval, pharmacokinetics, pharmacology. So it was nice to see not just the bench work, but the other side and how hard of a process it is. It's not, oh, we have a discovery, now we're going to put it in humans. I mean, there's so many steps between the discoveries of research and the application and patients. And then from there, since I had more research experience, that's when I applied to my PhD program. So that's now while I'm here at the We have an incredibly long name, but UT MD Anderson Cancer Center, UT Health Graduate School of Biomedical Sciences. It's a great program, but it's a collaborative between two schools, MD Anderson and UT Health. So that's why the name is so long. And so we have opportunities to work with PIs at either institution. And I went there because I swore I was doing cancer research, and I actually ended up in neuroinflammation. So I have a dual, it's like a dual degree, dual programs on my degree. So neuroscience is actually my first one, since I was like, I need more training in neuroscience than immunology, but I'm still in a lab that's mainly immunology and neuroscience. It's just by happenstance because it is a brain focused group. But we do a lot of like inflammatory work there.
So what year are you in your program?
I am a wonderful 5th year in my program. So I'm hoping to graduate soon. Within, I guess, it has to be within the next 2 years because my NIH fellowship will be over by then, which I was just awarded. So once I run out of funding, I definitely have to get out. So
congratulations.
Yeah. So your research must be really moving at this point. So how do you relate these ILC2s and the brain?
So what I'm looking at is the communication between microglia, which is the brain resident macrophage, and innate lymphoid cells, particularly in aging. Since there's been kind of a couple papers that have hit on ILC2 aging phenotypes, but also the differences between sex and how that plays with the immune system. So my leading hypothesis right now is that the ILC2s aren't the ones that are deficient. It still is the microglia. So is there a way that we can enhance the function of ILC2s way down the line, of course, but enhancing the function of ILC2s so that we can make the macrophages or the microglia still efficient at their cleaning up and less inflammatory, particularly for things like stroke and neurodegenerative diseases. And so aging is one of the main, I forgot what the word is now, but one of the main markers when it comes to the disease state of really any neurodegenerative disorder. But then also the sex differences, because we've already seen that up to about age 65, women don't really have strokes, but if they do, they're less destructive than men. But after 65, women end up being more disabled after they have their strokes. So understanding some of these immune mechanisms could be really helpful. And also when it comes to assigning therapeutics, there may be differences between males and females, which we did have some preliminary data for out of our lab about how in some cases women actually need more of a therapeutic to get some type of therapeutic effect than men do. So sex differences, biological sex is really important. Hormones are really important. But then, of course, how is this interplaying with aging as well?
Wow. Well, this is really you're coming together on a lot of different things that are impacting the pandemic, right? So age over 65, sex differences. Do we know anything about COVID and innate lymphoid cells? Has that, I don't know if
There was one paper that came out, but it had to do with blood ILCs. It didn't have to do with any like tissues or anything like that.
Sure.
So there hasn't been too much coming out. I keep trying to ask my advisors because we have a biobank. If I can do some fun experiments with it. And they're still a little bit leery because they're working on another project right now that's more essential. So if there's anything left over, they'll let me play with the human cells.
That's awesome. So Joelle, we want approximately 10 minutes per person or 15 or what do you think?
Yeah, I think that's good format, but I mean,
just let it go.
If
people want to go,
share, I don't know. It depends also on you, but
we're fine. I just want to make sure because I'm kind of keeping a little timekeeper to make sure that everyone gets the same amount of time. For Alexis,
let me ask Alexis. So, how important have been mentors in giving you confidence that you could do it?
Oh, they have been absolutely vital. Even in undergrad, I mean, the only reason I'm in research is because one of my mentors, it was actually my OChem professor. I was asking him for a letter of recommendation because I was going to do pre-med. And he was like, "Are you sure?" He's like, "You have the mind of a researcher. I think you would thrive much better in research." And then even how I got into research was, again, one of my professors had noticed I had more of a knack for research. So especially in undergrad, a lot of people set a great stage for me to be where I am, and continue to check in on me, amplify my voice, advocate for me. My master's degree, of course, was amazing. My PI there, he was awesome, always very patient with me. When it takes 6 months to recreate something, you have to go in every day. I screwed up sometimes and I would just come in very defeated. And he's like, it's okay, that's science. You have tomorrow. It's okay, go home. So that was wonderful. And then even now, my mentors now, they pretty much support, the sky's the limit. They let me really put my own hands. This is my own independent project. Nobody in my lab is really working with ILCs. So they said, find something that interests you, and you'll love your science, and just take it with you. So they've been pivotal in just, keeping me encouraged, keeping me successful, and just, keeping on with the dream that I've always had of being a scientist. So.
That's awesome. That's great. Well, I'm curious, before we transition to another person, do you have any anecdote about being Black in academia? And, you can make that light or heavy, whatever you'd like. But do you have any?
Yeah, I definitely will do the more light version because I always think it's funny. So I have a daughter, and I go to her school, and I try to volunteer as much as I can. Before the pandemic, on the weekends, not on the weekends, but on Friday, I would walk the dog down. I would let her play after school. And so a lot of people think I'm her sister. Obviously, I'm young. But she's like, no, that's my mom. And so they want to know what I do, or they ask me what I do. And I'm like, oh, well, I'm a scientist. And so I'm like the cool mom now because I'm a scientist, so every time I go sit down, they're like, oh, that's Blake's mom. She's a scientist. So I always think that's really cool, because she has such an international school. Actually, at her school, there is no majority race. So that's really cool. But to know that they seem so excited and ask me so many questions. I'm like, what do you do? And how do you do it? It just makes me excited to know that they have seen some type of representation, because I never got that. And so I just like knowing that I'm having a positive impact, just doing what I love every day, on other generations, and helping them come up and find a new dream and be able to share whatever their great ideas are for science.
Awesome. That's great. Well, anybody have any other questions for Alexis? And we could, if not, move on to Joelle.
So I guess I have one quick question for Alexis. So Alexis, you mentioned how doing research and things like that sort of allowed you to live up to that dream that you had. When was the first time you were really interested in science?
Oh, from the jump. I wanted to be a geologist, actually, for the longest time. I actually still collect precious gemstones and rocks. They're all around my apartment. Funny story, we were in Italy, and we were climbing Mount Vesuvius, and there's a halfway point, and I saw a rock. And I was like, I want that rock. So I said, I have to go to the top or I can't get it. And so I bought it. And then my mom, because, the 50-pound limit, she's like, how are you going to bring that back to the States? And I was like, throw out all my clothes. But I always wanted to be a geologist. I remember doing the science fair in second grade, where I was trying to look at the properties of toothpaste and which one is the best cleaner. Nearly blinded myself with one of those, I don't think it was scope, but it had diamonds or something, and I had a microscope, and that was just not a good idea. But from there, it's just, cultivated to different fields. And I think it's cool. I don't think I would have ever thought of immunology. Honestly, if it wasn't for my best friends, I wouldn't be here. I would probably be in cell biology or somewhere else. So it's just been fun watching the development, even for my parents, from, like I said, from very, very young to now an adult, and a growing adult at that, of the things that I can do and what I continue to do.
Awesome. Well, tell your best friend thank you for us.
Yeah, I will say, I'm glad you're not a geologist. I'm glad you're an immunologist.
Awesome. All right. Well, we'll move on to Joelle.
Hey, it's amazing to hear all this about Alexis. It's funny because we've been working together now for months, But I realized that having this podcast is going to be a chance for all of us to know each other even more than that. I mean, we're having meetings every week, and we have a Slack channel, and we've been working on putting together the Black in Immuno Week, and we work as a collective, and we know some things about each other. But yeah, it's really exciting that we're gonna get to actually know even more. So yeah, that's really cool to hear about Alexis, and I'm excited to hear about the other one as well, actually.
Great. So I think we're doing the round robin of ask what type of immune cell you are. I don't know if that means what your favorite one is, but at least what you represent.
Yeah, I think my favorite one is also the cell type that I am. I am a dendritic cell, and I think right away, let me say that they're the best cells in the immune system.
I think we're gonna have to have a fight here.
Yeah, there might be a fight. We might have to fight. We'll see.
I knew that was gonna, brush some people in the wrong way. I knew it. Too easy, really easy.
I'm not gonna fight with you about that.
Okay, I know you have so many pseudopodia. You have so many arms. You'd be maybe hard to beat.
Yeah, right. Yeah, it's like you really have to go really strong to fight the dendritic cell. I'm a dendritic cell. I have been traveling long distances to get here. I like to form strong connections. And it's all about, the Black Immuno Collective is creating these interactions and connections, getting things started, right? So, definitely a dendritic cell. I have some anecdotes about dendritic cell. They're really cool. I mean, they can break barriers, right? And that is something that we're trying to do, I guess, as a collective, trying to break the barriers that are limiting progression of Black people in academia. So the dendritic cells are doing that all the time, right? They're using a set of enzymes to break those extracellular matrices, and they're really, really strong, very impressive type of cells.
Awesome. Well, I think that there's probably a lot of people, we talk about dendritic cells a lot on this podcast. So there's probably a lot of audience members who can relate to you on that. And I think that dendritic cell won the Nobel Prize in 2011.
11.
12.
Oh, was it 11?
Yeah. Okay.
I think so. Yeah, definitely. Yeah, that was a victory.
That was a victory. Yeah. So you got that under your belt. So it's hard to mess with that. So what has been your journey so far? What's brought you from, whenever you first got interested in science to where you are now as a postdoc at UCSF?
Yeah. So the first time I got interested in science, I think it was in school. I guess I should say that I grew up in France. Maybe my accent has already mentioned that for me, but I grew up in France and in the area around Paris. And from parents that immigrated from French Caribbean island, Martinique, and they immigrated during Exodus. So most of my uncles and aunts also immigrated at the same time. Anyway, but there was no school for them, or it was very basic. So they immigrated and they put us in school, and that's where I think I got my first excitement for science and as a first gen to go to school, first gen to go to high school, actually, when you come home and you want to talk about these things that you've learned, your parents are like, yeah, okay. They don't necessarily understand. They see that you're excited. They don't necessarily understand. I have this memory of trying to explain some stuff, and it was not easy, but I have these memories of probably, in middle school, being very excited about science in general. And then initially, I think I was more interested in medicine school. I was probably more seeing myself as a doctor. And actually studying for med school, I found this huge book of immunology. And I couldn't do something else. I was just going through it, and I was just studying immunology. So I think it's the moment where I really fell in love with immunology. And so I actually forget about, medicine school and then got a master's in immunology, went to a grad program at Paris Descartes University, and and I've been in a lab under the supervision of Loredana Saveanu, working on dendritic cells and and studying out trafficking, the intracellular trafficking in the endosomes of toll-like receptors is regulated, and how the traffic and how the compartmentalization of these endosomes helps to control the immune response that is initiated by dendritic cells.
So that sounds super exciting. I feel like I could ask you some questions about that all day. But what types of things are you working on now? And sort of where's your research going right now?
Yeah. So I have currently multiple translational projects where I look at the immune system of patients, of healthy patients and healthy individuals. And basically all my projects basically converge into one single question. What are the factors that influence immunotherapy responsiveness? And one of the factors that I am particularly investigating, is the microbiome and the metabolome produced by the microbiome. And everybody has a different microbiome. So I want to understand how these individual differences may influence the immune system. And especially in patients where we're trying to use the immune system as a therapeutic strategy to fight the disease, right? And we've seen that, some people are responding to this immune modulation, some people are not responding. And recently, in the last 5 to 10 years, there have been some investigations that have shown that, if you have the responders have a different microbiome than the non-responders in some clinical contexts. And this is something that I want to dig into other clinical contexts as well. So I developed a project in cancer. I have another one in autoimmunity, one in transplantation. Basically, all these clinical contexts where we're trying to modulate the immune system, either to tune it down or to try to boost it to try to resolve the disease.
So how are you actually measuring some of those things? Sorry, Cindy.
Well, I was going to say, you could be delivering my talk. I always say, because I also work on the intracellular TLRs and talking about tuning up and tuning down and using that, how do we understand when they go wrong for autoimmunity and, under lower levels of signaling that lead to inability to, develop appropriate immune responses. You're right down my alley. I mean, your dendritic cells and the TLRs. I love it. I love it. I love it.
If we tweak the cells and we tweak exactly the receptors, get them to be DNA receptors, then I'm on board too.
You guys should have the TLR I work on is the DNA receptor, right?
Yeah.
Perfect.
You guys should have a separate Zoom meeting and geek out over all that.
Oh yeah,
that sounds really fun.
We can definitely talk offline. Yeah, I'm very excited to chat with you about all this. Yeah,
love it.
And it's funny because there is this connection between my grad research work with toll-like receptors and endosomal trafficking and all that. That is very basic science, and now I've moved into more systems analysis. So I'm basically using single cell methods like CyTOF, single cell RNA-seq, to analyze the immune system. But I also collect microbiome samples from stools and other parts of the body to look at the microbiome and also the microbiome-derived metabolome. So using mass spec and shotgun metagenomic. And so I've grown my computational skills up, and I'm pretty far from what I was doing during my grad research. That was more like biochemistry, confocal microscopy, but I'm planning in the future to merge these two things. So yeah.
So speaking of the future, you're a postdoc, so you must be thinking about what the future entails.
Yeah, it's been multiple years that I'm postdocing, but right now at UCSF it's been 3 years, and I'm planning to try to wrap things up within a year. I'm excited to at some point be on the market. I think I'm targeting to join faculty somewhere, someday. So,
Okay, well, maybe you'll get the immune bump and it'll put you on people's radar when you go out into the market.
I hope so.
I hope so.
I hope so.
Oh yeah, that'd be great. So I actually had a question for you because you mentioned that you were first gen, and you actually mentioned you were the first year family to go to high school. And I kind of want to do a poll. Like, how many of us are first gen?
First gen what?
First gen what?
First gen college?
I'm
First
a
gen
first
doing
generation
advanced
American.
degree?
I'm a first generation American.
That you can say, yeah, that's
So I think, most of us are either first gen to go to college or first gen to go to college and first gen go to advanced degree, or at least just advanced degree. And I think that's telling, because there's a lot of people out there wondering, is this for me? Can I do this? I think we can all say, yes, you can.
Yeah, definitely. And it's part of the mission of Black in Immuno. I think we definitely want to convey that we exist and we're doing awesome science. And we definitely want the generation coming to be able to refer to these people and being like, whoa, they do these really cool things. Maybe I can go into that as well. And we want to create this excitement in the generation that are coming. So yeah, it's part of the mission, definitely.
So do you have a story or anecdote about being Black in immunology?
Yeah, I do have good ones and I mean positive and negatives one. I guess one really good classic is when I talk about my science, or when I've been talking about my science. I think some people would be like, oh, wow, you're so articulate. Like it's a surprise, and I think it's a big classic. Actually, I didn't know that it was such a classic, but reading the thread Black in the Ivory on social media, I realized that, yeah, a lot of people have had exactly the same sentence with exactly the same look. And yeah, it's part of the things that we encounter, unfortunately. But positive stuff as well. I think, my grad student advisor, or my grad school advisor, or my PhD advisor, Loredana, she is a very smart lady, and not only like lab smart, but also, life smart or street smart. I don't know. And I think, I started with her, and I think she really fast got what it means, or she really, I think she really fast or quickly got it, what it is to be Black, because she would notice that people would act differently around me or have some sort of behaviors, and she would be pissed. And she would be, we would have this conversation, and she would be like, I noticed, I've seen that, and just this acknowledgement was, I think it was really something really good for me, because I felt like I was seen, and I think it's very important, right? So I could go through a lot of things, because at least my PhD advisor was seeing me, and she was seeing what's going on around, and she was in some cases protecting me, and in some cases she would speak out against some people. And so, it has been really good to have this support from my supervisor, and she's been amazing. I mean, she taught me so many things. So yeah, I think, I consider my PhD advisor as an ally. I think, and I think it's good to have an ally on your side when you're going through grad school.
So far, we're 2 for 2 with you and Alexis talking about the importance of mentors. And so you're making me really want to make sure I up my mentoring game.
Yep.
Yeah, there are many opportunities right now, and especially since the COVID situation and the lockdown, there have been a lot of programs that are proposing to pair people, and many very interesting programs have come up in the last 6 months. And I think there is a lot of opportunity out there to mentor Black individuals, students. I encourage anyone to look it up. There are a lot of opportunities.
I think one of the things you said, though, was really important. And that's just acknowledging things when they happen, right? Don't just try and pretend it didn't happen, and that, let me make things better or whatever, but calling it out, speaking it,
yes, especially because
people feel that they're heard.
Yes, that. And it's like, you all need to, use your energy to be doing your science. You shouldn't have to be using your energy to explain to people why you're even there, or, yes, I speak fine. Like, what, all of this, it's just so much energy that you've just spent when you're like, I need to think about ILCs and dendritic cells. I don't have time to be worrying about, things like that. So I think mentors and PIs especially can really help deflect some of that, in acknowledging what the stories you said. So,
well, and even peers.
yes, yes.
We as allies, we as colleagues can be like, no, that's not okay.
I think I think your point though of saying that your mentor was female and that she was really good at that. I personally, as a female in academia, can understand and relate to that. That things happen to me, and I wish somebody would speak out and defend me. Right. So I think that we can see it really easily when it happens. But I think that we, as women in academia, also have to be strong enough to stand up and say something as well, and that could be a challenge for some.
Right.
Yeah, I guess experiencing some discrimination as women can definitely help to have some awareness. But there are also cases where it's not enough to really see things that are not in the same set of attacks and aggressions. And so even at that stage, I think I've read a text that was really good about it, but I forgot. Undoing the knapsack or something like that. It was sorry. But it was really
it's fine.
good.
We can if you remember it or find it, we can swing back to you for sure.
Okay. Yeah. Okay, sounds good.
So any other questions for Joelle before we move to, I think Elaine is our next person, correct? All right. Well, I think Elaine can take it away. We know what the first question is going to be.
Yes, I can finally talk about the real cells, the T cells.
Oh, come on.
Okay, Elaine, we are not fighting today. Like, we're not doing it.
Alexis and I know we have this, long-standing argument about, the real cell are the T cell. So I think my favorite cell, and I think I am also that particular cell, a CD4 T cell. And what I like about CD4 T cells is that they're very nice with the dendritic cells, so we tolerate them. But they are really good. CD4 T cells are really good at judging the environment and figure out what they need to be. So I really love that. I feel like it definitely fits my personality because a little bit like Joelle, I also moved around a lot. So I have to really adjust to different environments. So I like this ability to be like merge into the environment. So this plasticity of the CD4 T cell is something that I definitely relate to. And they're really nice. They're helping the immune system, to do what they're supposed to do. So I feel like they're like, keeping everyone in check and doing their job. So that's why they are the real self.
Do you have a specific subset?
I do study Th1, so the inflammatory type 1, but I think I like the Th17 because they are so popular in mucosal immunology and they're pretty cool. So I think I'm in between Th1 and Th17.
Well, they're both, pro-inflammatory. So I think there's a connection there.
That is also my personality. So
that's why we're always fighting.
I love it. Love it. That's awesome. So Elaine, what's your story, your journey?
Oof. Oh my God. My journey. So I was fortunate to have parents with PhDs. So my parents are scientists, but plant scientists. So growing up, my dad would take us to the garden and always trying to plant something new. And I don't like nature. So it was pretty, it was hard growing up and then, having always to think about plants and touch things with your hands. And this is disgusting. But I did it because I love my dad. So I was like, I'll do it for you. But I think growing up in that environment, really pushed me to be curious, to always question things around me. So I think at heart, I was a kind of a scientist already because I was very, very curious. And I had the opportunity to really always express myself growing up. So I grew up in the Africas. So I guess I was born in Florida and my parents moved to the US for their PhDs. So they were at the University of Florida. And then after that, my dad was like, he doesn't want to stay in academia, so he's gonna go back to Ivory Coast. So we all went back to the Ivory Coast, and over there, I felt like I was really exposed to, obviously because it's a tropical area, was really exposed to like viral diseases or like just pathogens in general. So like malaria was the thing. Like I had malaria multiple times. And funny enough, my younger brother, like almost never had malaria. It was like super resistant. And I think that's something that really bothered me as a child. It was like, why are you not sick? And I always get sick. I was very like the weak one in the family, I think. So like I was really frustrated with my immune system. So I feel like that already like kind of made me more curious about the immune system. So why am I so weak and why is this boy so strong? So that kind of basically like made me more and more curious trying to figure out what about immunology, mostly about malaria. So I always like read about malaria, even though I don't study it, because I feel like it is my first love of like science, real like scientific research. So then I moved back to the US for college. So when I was 16, so I went to Temple University. I studied biology, and there I found a very good mentor, Dr. Jacqueline Tanaka. I think she's at Yale now. She was my professor in biology, and she was amazing. She listened to me because I felt like when I moved to the US, I became like a story. So I feel like I lost my individuality. Like I grew up in an environment where everyone looked like me. So I feel like I could stand out as me, the individual. And when I moved to the US, I feel like I became just like a Black person in the classroom. And like, mind you, I went to Temple University, which was one of the like most diverse university in the US. But like in the science classes, I was always one of. So I became like the representation of an entire group. So that was really scary. So when I met Dr. Tanaka, I feel like she was the first one to ask me questions about myself. And I was like, oh, wow, you do care about me. So that was really interesting. And also as a child of African parents, I was like, I'm going to be a doctor because obviously that's what you do when you're African parents. You don't want to disgrace your parents, so you're going to be a doctor. So I was pre-med as well. I was really like going to the pre-med route, and I started like doing research in the lab. That's every pre-med role. And I like the lab more than, going to the classes. So I was like, maybe I will explore that option. And I have really fabulous mentors that really helped me, guide me through the research process as well. So kind of like why I decided to go to grad school. So I took a year off before grad school. So I did a prep program with the NIH at Indiana University. So I was still in that phase, like, should I do med school? Should I do grad school? So I feel like that year was really good for me to figure out where the research was for me. And I actually studied microglia a little bit like Alexis does now. And I think I was really like in love with microglia because they're really good. Even though like adjacent macrophages, adjacent dendritic cell, I still like them enough. So they're like always, I studied microglia in the context of Alzheimer. So I was really like exposed now to like cytokines and inflammation. And I really like those concepts more than the neuroscience part of the project. So after that, I decided just to apply to like immunology programs. And that's how I decided to go to University of Chicago.
That's awesome. So what are you working on there?
That's interesting. So, my project is like a mix of virology and immunology.
Yay!
Brian's jumping up and down right now.
Yeah, I actually
I love the arms.
I actually started listening to TWIV to learn virology when I started grad school, because I was like, I know nothing about viruses, and I'm in this lab that study viruses. So I need to like, listen to what virologists think about, or like how they phrase questions. So I will listen to TWIV. So shout out to TWIV.
Wow. Look at
so my project worked on reovirus, which is a double-stranded RNA virus. So we study how reovirus basically change the immune response to food antigen. So we have like 2 strains of reovirus that I work with, and our collaborators at the University of Pittsburgh, so Dr. Terry Dermody. So they basically ship us virus. So I don't do a lot of virus manipulation. They just deliver them to me. So shout out to all of them, to them for all the virus work. So what I do, basically, I use a mouse model to study how this virus basically changed the T-cell response to food. And we believe that this is a trigger for like celiac disease. So we use as a model for celiac disease.
You think viruses are involved
sorry,
in human celiac disease?
Yes. I remember that paper on Twitter. I do. That was the paper. I am following up on that project.
Oh, wow.
Yeah. So basically, what we found in humans, so that people that develop celiac disease have high viral titer. So viral infection has always been associated with disease onset. So one of the best epidemiological study that was done was when they followed up children that are genetically predisposed to have celiac. So you need to have, in order to develop celiac, you need to have the DQ2, DQ8, which are the MHC molecules, so that the molecule that will actually present the food antigen. So what we found was that the incidence for developing the disease was higher with children that have higher viral titer. So there was more of a correlation evidence. So for my project, we are basically the first mouse model to show that if you infect mice with reovirus, you actually can change the immune response, the T cell response to the food. And in humans, you actually see this T cell response of people that have celiac.
So you mentioned earlier that you were sort of thinking about Th1 cells, specifically out of CD4 T cells. So are the Th1 cells kind of the pathogenic type of cell in this model?
Yes. So that's why I love them. They don't do what they're supposed to do. So normally when you basically feed a mouse with an antigen, like food antigen in the gut, what you get is a regulatory T cell response. And that's why we also believe that we also have the same response in humans. But what happened in celiac, or when you have infection with reovirus, you have this Th1 response. What is really interesting about the reovirus story is that the response to the virus itself, so the CD8 response, is similar regardless of what virus you basically infect the mice with. But only specific viruses can change the immune response to food. And that's really what my project is trying to identify. So like the question we ask is basically like, what does this virus either impacting dendritic cells in this different way? So in the gut, we have a kind of a hierarchy of dendritic cells. So some are really good at monitoring the response to food. Some are really good at monitoring response to virus. So we think that these viruses are able to like impact the dendritic cell differently. So depending on the dendritic cell you are impacting, you are actually like changing the immune response either to food or to the virus itself.
That's awesome. Yeah, I think that maybe even, I don't know if you're looking at the microbiome, but it seems like something Joelle and you could collaborate on, because I'm imagining there are going to be changes. And of course, microbiome could also mean viruses and bacteria, but I guess looking at bacteria as well.
Oh, yeah. I actually did look at the microbiome, and I'm hoping somebody else in the lab takes the project.
You're like, I have no time for this.
But I did look at the microbiome because I basically studied 2 strains of reovirus. So one strain actually changed the immune response to food and the other doesn't, even though both strains have an antiviral response. So the virus that doesn't induce the immune response to food, actually in germ-free mice is able to do it. So it means that under the SPF condition, the microbiome is actually suppressing the ability of this virus to do it. And somebody needs to continue that.
You're like, I gotta graduate.
It sounds very interesting, though.
Yeah.
It seems really cool.
Yeah, I like it. I feel like my project really allowed me to be like a fake virologist and like a wannabe immunologist. I'm in between, so I love that.
So what?
I'm listening to this and I'm like, I want to ask you 1,000,001 questions about the science, but I want to hear a little bit more about what it's like to be Black in academia from you. And I actually also had 2 questions. That was one, but the other one was, you mentioned a prep program, and I wonder how important was that in your development towards going to graduate school, and what was the most useful component of that?
Okay, I can answer the prep program first because that's the easiest. I feel like without the prep program, I wouldn't have been in grad school. Because I was always pre-med, so like up to my 3rd year, and that's when I started doing like research. So I was already late in the research game to be competitive for graduate school. So the prep program really allowed me to have this additional research exposure. But most importantly, the program taught me how to know myself better, because I felt like I had to rotate in labs, which I've never, because in undergrad, basically you get what you get. So like in the prep program, I had to choose labs. So I had to choose how do I learn? Like what kind of mentorship do I want? Those are questions I never asked myself in undergrad. So the program gave me this 1 year of figuring it out before trying to figure it out in grad school. So I had the opportunity to rotate, choose mentors, ask them questions. Are you going to support me? Are you going to help me with presentation? Like even learning how to present with PowerPoint, something that I've not learned a lot in undergrad. I was able to do that in my prep program. So I came basically more prepared in grad school and more confident. So like the prep program basically like boosts me up to be ready for graduate school, but also exposed me to a lot of reality of graduate school, because basically that also goes to my anecdote. I had a very bad experience starting my prep program. So the first lab I was in was super racist. And I think that definitely broke my spirit a little bit, because I still think about that when I sleep. So the mentor I had was really, really harsh. And I think she told me basically like, I need to be a medical doctor because I can think like a scientist. And then it was a compliment. I was like, it sounds like a compliment, but it's not really a compliment. I don't know. But that definitely affected me. And I had to switch. So basically the prep director basically put me in a different lab, because obviously I couldn't stay there. But that was like a real wake-up call for me, realizing that everybody is going to support me. Not everybody will be for me, even though people have good intention. I think in general, she was a good mentor. She was like training and supporting, but just not me. So I feel like that was like a wake-up call for me, that I have to actively look for people that will support me. And I kind of have to learn that, yes, I have people that will be against me and I need to be okay with it. And that doesn't necessarily reflect on my ability to be a scientist, basically a version of themselves. So that was an experience for me.
Great. And just the thought that like there's one way to think like a scientist, and that she could identify that in a prep program student. I mean, that's just so crazy to me. To even say that out loud is just so embarrassing. So I'm sorry that happened to you.
Yeah, but I think that what you said was just, it was so mature and so fantastic. And I think it's worth repeating, is just that, like one person is not the perfect match, right? And if you're in a lab or you're in a PhD program and it doesn't feel right, and you're not clicking and you don't feel like you're getting the right support, think about a different spot. I did that in my PhD. I was in a lab and I was extremely unhappy. And third year I switched. And, it all turned out okay, but I was terrified. I was terrified to switch labs. You think it must be me. It can't be the mentor. They're where they are. They're on this pedestal and you just, look at them. But to have that, knowledge about yourself and what you needed to be successful and how that program helped you, that's amazing. It's a great story. I love it.
Yeah. I also had like a very great cohort of friends in my prep program. So I feel like I doubted myself, to be honest, because I was like, well, maybe it's me. Like, it's not the person. So like me sharing those feelings with people around me, they're like, no, it's not you. You should tell somebody about it, do something about it. And I did, because they really encouraged me. And when I switched lab, it was like day and night. Like the mentor that I had with the second lab, like up to today, we are still in contact. She was still like, hello, Leary, shout out to you. She would always like email me, text me. She's already thinking about my postdoc lab. And I was like, I'm not even thinking about that. I feel like she was a cheerleader in undergrad, and she brought that spirit in the lab and also make me always happy. So she always clapped. So when I go to lab, like I always also clap, because it makes me in a really good mood. I think she's amazing. So I feel like switching was definitely good. Showed me like, there's better somewhere else. So.
That's awesome. Well, I love your stories are great. Loved hearing about your science. Does anybody else have any questions before we move on to Justine? All right. Thank you, Elaine.
If I started asking about the virology, we'd be here for a really
I know.
long time.
I'm like, I have to hold back science. We're going to have people back to talk about this.
Yeah, honestly.
We're thinking about all these really interesting topics we can bring up on Immune later.
Yeah, no, I love that. I'm thinking throughout the 2021, we just have each one of you back. You pick a paper. We'll go through it. We'll talk science. More
awesome.
Be great.
Awesome.
All right. Well, Justine is up, and she can tell us all about herself and what immune cell she is.
So I'll start off with my immune cell. I would say I'm a macrophage.
I'll do the arms now.
And just jumping.
I like food. I like to cook. It's a past hobby that I did a lot with my family. I'm a first generation student. My family is from the Caribbean. When they came here and they had me, and I went to undergrad at Stony Brook University in New York City. That was my first real exposure to research, I would say. I was a pre-med like Elaine said, I came from a family where you're successful if you're a doctor or a lawyer. So I didn't even know that getting a PhD or being a scientist or a researcher was even a thing until I would say my senior year in undergrad. And thankfully, I had a very, very great advisor, Howard Sorokin, in the neuroscience department. So in his lab during my last year, I did a lot of research on neural development, particularly on the FGF signaling pathway. And I identified this protein called Churchill, used zinc finger nucleases to do like site-directed neogenesis. And we found out that Churchill was involved in cell migration and neural development, or the mesodermal development. So that kind of like really jump-started my interest into research, just being able to ask a question and then coming up with different experiments to answer that question excited me so much. But I didn't have enough research experience at that time to directly go into graduate school. So I did the same program actually as Elaine. I didn't even know that she was
wow.
I was a prep scholar as well at the Icahn School of Medicine. And that's when I joined the microbiology department, and I worked on identifying different proteins that were involved in the transcriptional response of the antiviral response to influenza. So that was like a great experience because I had the time to actually be taught like molecular biology. I in my previous lab in neuroscience, I kind of was doing a lot of development, behavioral studies. So this was my first hands-on experience, like running a Western blot or things of that nature. So I felt like that gave me like a really strong foundation, and I really loved the environment at Mount Sinai. So I just transitioned from prep to the PhD program, and I did various rotations, and then I ended up in my lab now, which is Cecilia Barron's lab. And right now I am looking at different prenatal environmental exposures and how they are connected to offspring's allergic disease risk.
Oh my gosh. Just hearing that is like beautiful sounds to my ears.
For anyone who is not watching, because we do the video on YouTube, the way Steph's eyes lit up at the word prenatal was pretty impressive.
I'm just like melting over here. Scientific heartthrob. Anyways, go ahead.
Talk a little bit about it. So right now I have developed like a bunch of in vitro assays to look at the immune response in children that are at risk for developing allergic diseases. And eventually I will move on. Once I finish validating some of my preliminary experiments, we have 2 birth cohorts that I would use to validate the information that I'm presenting or generating right now. So my 2 birth cohorts are 1, prenatal stress, so how psychological or chronic stress during pregnancy alters the children's immune potential and how that can potentially skew the CD4 responses to a more heightened Th2 response and potentially lead them to be more susceptible to develop allergic diseases. And the second cohort that I'm looking at is more on the farm exposure side. So farm exposure has been shown to play a protective role in allergic disease development. So I'm really looking at like these 2 cohorts, the children's innate immune responses, because not much is known of allergies and innate immunity and how that initial response is generated. It's kind of thought that there's like this hyperinflammation and that could lead to a loss of central tolerance. So that's what I'm really like trying to tease out right now.
Oh, yeah, that's excellent. And so those cohorts, those are ones that you had your team enrolled in during your time, or was it a cohort you already had samples from?
So it's a mixture of both. So it's a collaboration with labs at Rochester for the farming cohort, and at Mount Sinai we have our collaboration, and we have our own cohort of inner city patients that come in, and they get their psychological scores based off of like surveys of like this crisis survey. So how much adversity you're going through during your pregnancy.
Sure. Sure. And I don't know if you have already completed enrollment on that, but I would imagine that COVID would play a large role. And I'm curious, the stress of a pandemic, are you thinking about that in terms of your studies?
Yeah, so we recently just restarted collecting samples. COVID put a pause on a lot of things.
Sure.
So I'm like indicating like these are my COVID samples in the sense. We'll see. We'll see once I finalize my current assays, because I do a lot of like flow cytometry, spectral flow cytometry. So once I finalize my panels and move on to like my clinical samples, yeah, we'll see.
That's awesome. And it highlights too, there's a lot of different model systems that immunologists use. So the audience has heard about mouse models, clinical cohorts looking at, can it translate to humans? And then I don't know if I think we're going to talk zebrafish. I thought I saw that on the notes somewhere. I don't know if somebody had a zebrafish model connection.
Oh, I think that's Medina.
It was Medina, I believe.
Yeah.
That's right. It's Medina. Yes.
Yeah.
And she's going to record later. Maybe we're going to slice it in. I don't know, beginning or end. We can chat about that. But yeah, so you can use many different types of models. So, but the clinical cohort one is great because the translational aspect, human immunology, we learn a lot about mouse immunology, but not as much as human.
What's in your future? What would you like to do next?
So I am planning to do a postdoc. I'm very interested in staying into academia. I feel very strongly that I wouldn't say you can't be what you don't see, but it's very helpful to see people that look like you and the positions that you're trying to attain to become. So yeah, that's my plan, to stay in academia, do a postdoc.
Awesome.
So do you have any anecdotes for us about what we should, be aware of about being Black in immunology?
I would say I had various experiences, good and bad. I don't view like the bad experience as like something negative. I view everything as like a learning experience of something I could learn from that. So I would say most of my experience have been like microaggressions, things of that nature. But what has really like helped me on a day-to-day basis is having a mentor that advocates for me. I think that's very important, to have someone that talks up for you when you're not in the room, and provides you with tons of opportunities that you may have not even known about. Like I said, I didn't know that this existed until probably like 8 years ago. So I have no idea what the resources are. I have no idea how to navigate. So it's very helpful to have multiple allies and advocates in the corner to help me grow during this process.
Yeah, absolutely. What for your postdoc, are you thinking of staying within the same field? Are you thinking of branching out? Do you have any thoughts on that?
Yeah, definitely want to stay in the maternal health aspect, but I'm keeping it very open. So my background in undergrad is neuroscience, and then I kind of did like this big switch in a sense to immuno. It would be very nice if I could merge those two together. I would say I'm a little bit lucky because my thesis project is in a sense like my dream project, like to study stress and how that impacts the maternal health, and then to see the potential impact I may have on their offspring or generations to come. To me, that's very interesting. So,
if you want to stay in New York, I bet Steph has a suggestion for you.
I do.
That was what I thought too. Yeah,
I do. Yeah, my current boss. So we're a pediatric maternal immunology lab. Sally Permar, she's actually moving to New York City to become the department chair of pediatrics at Weill Cornell. So if you're interested, now we study viruses that impact. So mostly CMV, HIV, but we're doing a whole host of different ones. So if you want to get in contact with her, I could give you that info.
That sounds amazing. I do have a little bit of viral working in the department at
Right. Well, and the great thing too about a postdoc, and I don't know what the group thinks, but there's the question of, do you diverge very far away from your graduate work? Do you not? Because the startup time to get those skills. But I think, picking up new skills can only be a benefit. I don't know what Cindy, Vincent, Brianne think, but
That was what I did, and I would definitely go that way again in terms of trying to learn something new, and learn new techniques and learn a new process, and also learn about how I learned about projects.
Yeah. Yes. So, Justine, do you have any other final thoughts or things you'd love to talk about? Oh,
I think we lost.
I think we lost your sound.
I think your mic disconnected because it doesn't say it's muted, but we don't hear you.
What we could do, Justine, think of whatever you wanted to say and hold on to it, and then we'll circle back. Okay, great. So I think that our last person is Yvonne, and we are going to let her take it away and talk about her immune cell of choice.
Okay, so it's interesting that you refer to me as her, because I was actually noticed in the beginning we didn't ask each other our pronouns.
Oh, yes.
I go by they, them. And that's another thing about diversity, you know. Having more people who identify differently than you makes you aware of those things that you may not have been aware to otherwise.
Absolutely. Yeah. And, just even bringing that up, taking it as a learning opportunity, we did not start the podcast doing that. But in the future we will. And do you have anything else, that you want to talk about in regards to expanding the definition of diversity? Not to put you on the spot, you don't have to, but.
No, for sure. Because also disabilities, whether it be visual or non-visual, is something that universities don't really consider when they say diversity. Most of the time they mean racial or ethnic diversity, but diversity with able people is also something that universities need to be more accessible to.
Absolutely. Absolutely. Great. Well, thanks for that. And your, immune cell type.
Yeah. So apparently me and Alexis have a lot in common, because I also said I was an ILC. They're very rare, but they're very niche and necessary in order to combat an immune response. I also studied type 2 immunity. So I would say ILC 2 as well. And they're very unique and they're interesting to study, and they're very niche in their environments. So I think I'd relate to that a lot.
That's awesome. So tell us then about your journey getting to where you are at now, and kind of what was your first moment you knew? I don't know if it was a moment, but when you knew science was for you.
Yeah, so I kind of always knew science was for me. I was really interested in biology in general. I didn't know it was called biology, but I was really interested in animal behavior. I thought animals were just so cool, because they're different from us, but there's things going on in there that we just can't access. So I was very interested in animals, and I wanted to study animals no matter what it was, from elementary school upwards. But I didn't want to be a doctor, and I didn't want to be a vet. So I was like, there's some way I'll get to science without doing those two things. And so then in undergrad, I was made aware of internship opportunities. And so immediately freshman year, I started applying to internships, and I got my first internship at Brandeis University, where I was in a research-based lab. And that's when I was like, okay, I can do research for extended amount of time without becoming bored to it. Because people who are pre-med and they go to research internship, they're like, okay, this is not for me. But I didn't have that moment. So I was like, yeah, I can do this for as long as it takes, basically. And then I had an internship every year after that. But where I became interested in immunology was my second internship at University of Virginia, where I was in Jan Kipnis's lab studying lymphatics in the brain and
nice.
The central nervous system. And
he's on Twitter, is that correct?
Yeah,
I was going to say, because I think I'm reading his tweets a lot, so yes.
Yeah. He recently moved to WashU, but he was at UVA when I interned there. And I got really interested in how immune cells can influence the central nervous system, or the nervous system in general. Because I thought I was wanting to do neuroscience, but it's a very complicated field. I wasn't understanding any of it. And so I was like, this is how I get my foot in the door with neuroscience. I can do neuroimmunology, even though immunology is just as complicated.
I was going to say, it didn't choose the easier one.
Yeah, but at least in my head, I could understand immunology because it's more clinically applicable. That's just how my mind works. But so then I got interested in immunology, and then I had another internship in neuroscience because I was trying to see if I still was interested in neuroscience specifically. And that's where I decided that I would go into immunology at UPenn, where I have my third internship. And that's how I ended up here. I'm actually on like the earliest stage in my career, I think, of everyone here. I'm a second year.
Nice. Are you pre or post prelim or candidacy rather?
Pre.
Sorry. Pre. Okay.
Pre.
Got it.
So I'm not a candidate, but I'm in the graduate group.
Got it.
Awesome. Well, good luck.
Mhm.
Yeah, good luck.
You'll do
Thank
great.
you.
Yeah.
Thank you,
So,
guys.
what types of things are you working on in that process?
Yeah. So after interning with Yoni, I was really interested in neuroimmunology. And so I knew I wanted to do immunology, but I still wanted to keep some neuroscience elements in there. So I'm currently working on a neuroimmunology project. I joined Jabrowski Herbert's lab. He's an immunologist here at Penn, the only Black faculty on staff. And but he was very open to me basically incentivizing this project from the ground up with a collaboration with Ishmael Abdus-Sabbar, who is a neuroscientist who did his biology PhD here at Penn.
Right.
And he's in the neuroscience program, I guess. And so I'm being co-mentored by them. And we're studying, he studies itch and pain sensory neurons and the peripheral system. And then Jabrowski Herbert studies type 2 immunity. So we're trying to combine those fields.
Wow.
It's very, very early stages of the project. But what I can tell you about it is that we are injecting or use optogenetic tools to study sensory neuronal behavioral outputs in the hindpaw of mice. And what I would like to do is to identify the immunophenotype and how neurons are influencing how immune cells respond to inflammation.
Wow.
There's some investigations that suggest that there's primarily type 17 immune response when you optogenically stimulate neurons. So somehow neurons are influencing how the immune cells are tailoring their response to inflammation. And that's what I want to study.
So is there interaction the other way? You mentioned how the neurons can influence immune cells. Do immune cells influence neurons in any way?
For sure. Neurons have a lot of cytokine receptors, such as IL-4. They also have a receptor ST2, which is for the inflammatory cytokine IL-33 released by epithelial cells. So there's a lot of avenues for neurons to respond to immune cells. But it's a really open field right now. It's kind of like Alexa said, there's not a lot out there. So it's really, really fun to study because you can pretty much do anything.
Yeah, there seems to be a lot of crossover too of the neuroscience interest, which it is a burgeoning field. I think there's a lot of areas that you can kind of cover and kind of develop your niche, which is integral for us as trainees. So that's very exciting.
Yeah, I just want to say, Yvonne, I think it's great that you're studying like neurons and the immune system, because that was actually one of 3 proposals I put together, even for mine, pitching to my PIs, which I guess when I was writing the stuff, I didn't realize how to write that. They're like, this is an R01, so pick one. So I ended up picking microglia. So it's cool that there's other people out there kind of doing the neuron track. So I think that's awesome. I'm so excited to see what you do with that.
I was really excited to hear you talking about it. I was like, oh my gosh, another one. We're out here.
We're here.
Awesome. I actually wrote my NSF about neuroimmunology, but I'm not doing that obviously, but it was like in the central nervous system with Toxoplasma gondii, which is like neurotropic virus. I was rotating Chris Hunter's lab. So I was wondering, because they, once they get to the brain, they can form these plaques and they just stay there. Invisible to the immune system. And so I was writing my NSF about investigating into how it's avoiding detection by the immune system.
It's going to be so cool to follow you guys for like the next 20, 30 years.
Yeah, I know.
Your careers go.
I know.
I'm going to put a PubMed notification on each one of you so it enters into my email.
So we've kind of been going and asking a couple of different things, and we've been asking everybody sort of some anecdote about their experience being Black in immunology. So what's yours?
Yeah. So I wouldn't say it's specifically to immunology, but my undergrad, I went to HBCU. For people who don't know, that's a historically Black college or university. So population's like 92% Black. And it was very it's kind of a niche hub of people. So the successes, the failures, you're all seeing Black people doing them. And it's really easy to fall into comfortableness with that. And so, but luckily for me, I had my internships where I would go out, extramural internships, where I'd see what the community of scientists actually looks like. So interacting with my peers on campus and then going out into these internships, it was starkly different. And clearly you could see the problem with the lack of diversity on these research-focused institutions. So I think that's what really brought up the advocacy in science specifically, because I was advocating in NAACP and things like that, but specifically in science, there needs to be changes as well.
Yeah. Can you speak more to that in regards to HBCUs and really this pool of talent scientifically that, there's not probably as you found out, not as many people from HBCUs that come into STEM. So what can we do as allies, as people who hope to run our own labs or are running our labs to really connect with that pool of excellent potential scientists?
For sure. Really just reaching out to them, having panels and things to advertise your grad schools and your programs. I know Penn recently did that, thanks, because I kind of pushed them to do it, but we recently did it at Hampton University, where we talked about the grad school program, the application process, what you can expect from grad school. A lot of people want to do pre-med, so advertising the MD, PhD, as well as the PhD is really important. Letting people know that you don't have to just pay for med school. You could get a PhD and have it paid for, and then have 2 degrees at the end of your studies. So it's really have to put your foot forward and reach out to these schools and let them know that you also want to access that pool of applicants potentially.
So yeah. Do you think that a potential grad school giving some information about how does the application process work and demystifying all that is a good strategy to help with opening up the application pool at both an HBCU, but for diverse undergraduates across the board? Is that something that grad schools should be focusing on?
For sure. Our Office of Research and Diversity, Equity and Inclusion Director, he does a lot of work in Puerto Rico. So he reaches out to those universities there, and he gets in contact with them and has these panels to invite their applicants to apply to not only the PhD programs, MD PhD programs, but also the internship opportunities they have here.
Fantastic. Yeah, I think that, not to put you on the spot, but I think that that's an aspect of diversity that we don't always think about in terms of undergraduate institutions. So anything else, Yvonne, that you would like to talk about, touch on? And then I think the next part we'll talk a little bit more about the Black and Immuno Week.
No, yeah, let's get to Black and Immuno.
Oh wait, can I ask them one more question? Yeah. So when did you first realize that you were interested in science? What sort of got you into science in the first place?
Okay, so I was really just interested in animals in general. I didn't want to be a vet, so I was like, how do I study animals without being a veterinarian? And eventually I told myself I was going to be a zoologist, not knowing what that meant. But trying to follow that path to be a zoologist, I just did whatever sciencey thing came my way. And then in undergrad, I realized that you can go to grad school without having to go to MD or vet school. And you can do masters or prep programs, postdocs, or PhD. I didn't even know you could apply to a PhD directly until I got to undergrad. But that's how I went from wanting to do science to doing it.
Well, I did want to swing back around to Justine. I don't know if your audio's back.
Can you hear me?
Yes.
Okay. Perfect.
So I didn't know if you had anything else to say. I didn't mean to cut you off at the end there.
The only other thing that I would have to say is to promote Black and Immuno Week. Our week and our collective is all about amplifying voices, and this can be used as a resource for institutions to find Black talent and to recruit diverse speakers for seminars and for conferences and other panels. So
we hope
and
that
podcasts too.
Yeah, that would be great.
Is there going to be a roll call in the first day? Because I feel like those first days of some of the other weeks, my number of people I follow on Twitter has risen quite quickly.
Yeah, the first day there will be a roll call. So we'll all introduce ourselves, our paths in immunology. And from there, we have various other events. Our second day focuses on innate immunity. We'll have speakers who work in that field. They'll give small talks throughout the day, and then we'll have a panel discussion. So innate immunity, we kind of think of like early response. So we kind of view that and compare that to early career paths. So you'll hear from a lot of early career scientists, PhD students, undergrads, and postdocs about their experiences, their journeys. And feel free, I think Elaine, maybe you want to talk about the adaptive immune day.
Justine and I are like the planning gurus. So for the following day after the innate, we have adaptive, basically the same format in terms of like the talks. We are going to curate like 4 to 5 talks by excellent scientists. Black scientists, obviously. And then really in the afternoon, we are going to go with a panel about career, something that at least most of us in the collective are really interested in, looking at what is happening after the PhD, thinking about postdocs, either academia, thinking about industry. So we want to have like a panel that will give us like a big perspective on all the things that immunologists can do with a PhD. So really excited about that. We really like put a lot of thought into it. So we made sure we have representation across the world. So we are hoping to have panelists from the US, from the UK, everywhere in Europe, and also in Africa. So we want to highlight all Black immunologists, like the entire, we try and at least compensate a big part of the Black experience around the world. So we'll see how that goes.
And where could people find Black in Immuno?
Yeah, they can locate us at blackinimmuno.org. Our website is beautiful, so please take a look. All of our events will be there, as well as RSVPs for you to RSVPs for our events, and more details about what the hashtags and how to promote on each specific day.
So can you remind us what the week is? What the dates are?
Oh, yeah. So it's going to be November 22nd to 28th. So we are also like thinking of donation. So we want to support all the Black scientists. We really want to make it a point to show that we value the contribution of Black scientists. So we also want to compensate all our speakers, all the organizers, all the people that are putting work into this, into really amplifying the voices of Black scientists. So we definitely are looking for donations. So I think Joel and his team really did a lot of jobs on trying to curate a very like simple process for institution and individual to really support our mission.
Yeah. And also to add to that, we also are looking to create scholarship funds as well, or travel grants for up and coming participants, graduate students, undergrads looking to go to symposiums and to get out there and to learn more about their field and to interact and build their networks. So.
Yeah, I think that's what I really enjoy about what we've curated for Black and Immuno Week is that it's not just a week. It's not just a one-time thing. Even with our goals, we are aiming to file as a nonprofit, a 501c3 organization, so that we can continue to do awesome things like workshops and trainings. We've curated a list of different scholarships and funds that students can go and apply for at all careers. We're working on trying to do more educational outreach, even from the grade school level, so that we can have more exposure. Our website and our movement is not a one and done. We're really trying to shake up the immunology field and continue to be here, be loud and bring more people with us. Because honestly, I've had so much fun. I know even with the planning, one of the questions was, okay, now what Black immunologist are we going to bring to our panel? And I remember just like... about that. I can show up. I'm pretty cool, but I think we might want some other people to be there. So that was kind of one of our struggles at first. So we really did have to hit the internet, and I know we're out there. I feel like almost when one of my other things, I want to be an astronaut at some point. And so, I feel like we're sometimes calling out into the ether, like, is there other intelligent life out there? But we're there, and it's awesome. So every time we pull in somebody else for Black and Immuno, I'm just in awe of the talent that we have and just how much fun we have. So I'm excited to see what other things, and I know Elaine will get on me, but we are also trying to host some socials because she's all about the fun. So we do have a fun channel on our Slack that, we post, just fun things, usually our song of the week or theme of the week, which is always great because then, we're all kind of on the same page. So this is more than just a one-off thing. Always check back on our website, our social media handles. We're on Twitter, Instagram, and TikTok. So we're just going to have fun with this and just keep the communication going.
So I was looking at your schedule here, and there's one thing here that I'm curious about. Immuno talent show.
Oh, wow.
What are you going to do?
So I think that for that day, we really wanted to highlight the things that immunologists do that are not immunology. So we have a lot of talent already in our collective, people into cinema, people with like graphic design skills. So we really want to bring all those skills like to the forefront. It's like you're a full person on top of being an immunologist. So you have all this other, passion that you are going through basically.
That's great.
Fantastic.
Yeah, definitely shout out to Yvonne because she did quite a few of the graphic designs that we have.
Oh, cool.
I think she put up the one there. Is that your symbol?
She didn't do that logo, but on the website, the one where it's the Black immunologist with the pipette and the immune, like every time I see something that comes from the graphic design team, I'm like, oh, bless you. It's so beautiful. I just want to like print it out and, put it on the wall. There's one of our graphic designers. He really likes more cartoony type things. So I'm hoping that we can get a shirt pretty soon because the way he did the macrophage was just, I was like, most of us were like, we identify with that cell. So we're going to have merch. We're going to make this a big thing, like I said. And it's wonderful because when I first kind of came into the collective, because that was kind of where I was like, we need a 503c. We need to collaborate with other Black and Immuno. But I like to feel things out at first. And just to see everybody was like, no, we need to do this. Like, go big or go home. And like, that's just been so encouraging that, just dream bigger. That's what we're doing every day. And every day that we come together and work on this, just dreaming bigger and bigger.
Yeah, it's amazing how the team came together with like different skill set. And I feel like I'm always shout out to Yvonne because they have done amazing thing. I think like the banner was designed in like less than a week. And I was just like, I have no graphic design skills, so I can appreciate that. And I really like that we definitely split into like our strengths, what you're really good at, kind of advocate for the group in different capacities and everything just came together beautifully. I think that most of us haven't done this kind of work in terms of like planning this week-long event, of planning like this organization for like the future, very long term. So I think this was really like an interesting experience that we started our first Zoom call not knowing each other. It's like 6 strangers coming on the screen and like we only talked on Twitter and it's just like, okay, you're going to meet those Twitter strangers now. And I think and it was a really interesting experience. So now I can always, make fun of the ILCs and I love that. So I love the opportunity.
It kind of sounds like all the different cells of the immune system coming together with their strengths to
Yes, absolutely. It's actually kind of funny because with Joel, I actually met his mentor in his lab last year at my university. We have a Kopchik symposium for the Kopchik fellows, and we had invited Dr. Spitzer to come speak at our symposium. So I had I had dinner with him. I don't know how much of his presentation was Joel's work, but I remember just being in awe of that work. And then when we got connected on Twitter, I was like, dude, I know you by tangent. This is awesome. So that's also been really cool of just like, oh, okay. Like, it's a smaller world than what you think. So it's just been so much fun. I feel so much better after we have our meetings, just being on the Slack. Yvonne, they talked about, disability, and I'm kind of going through that right now, and like worsening disability. And so, I can still just kind of be, today I told them it was a rough day. I was going to come in my grumpy cat onesie, but I changed my mind. But, just the encouragement that we get each and every day, the gifts and the memes, it's a wonderful place to be. And I just feel so much more empowered and just so thankful for these Black-in weeks, because I feel like they are a very empowering set of movements. I also get to be part of Black in Neuro, yay for neuroinflammation. So I get to see both sides of Black excellence and how we are marrying these things and how much work Black and X are doing with each other and the collaborations that we're putting together. So I'm just excited to see what these movements do for science and for academia in the years to come. Like, I'm so excited. It makes me so happy to think about it for the future.
So I have one question. Oh, go ahead.
I'm honestly thinking to add on what Alexis just said, that this is going to change how we see being a scientist. All these movements, I think they're really going to change how we can approach the vision of what is a scientist, because it has given so much visibility to a lot of Black researchers, and actually also help us to connect, as many of us have said already. It came together just because we were looking for other immunologists, Black immunologists, to connect with in the first place and to amplify. And we've started to cheer ourselves, each other, on Twitter, and being like, okay, let's do the Black in Immuno Week. Let's do it. And I think, I remember Justine pushing me around, and then I was like, okay, let's do it. And I think Elaine came in and sent some tweets as well, being like, we're at here, let's do it. And Yvonne joining. So yeah, it has been very organic, the way that we've formed this collective, and very exciting. And as Alexis said, every time we're meeting, when we come out of the meeting, I mean, we're not exhausted. We're like re-energized, and we go into the planning, and it's really fantastic. And I hope that it's going to have an impact, and I hope that it's going to generate a community, not only us, the collective, but the community of Black immunologists. So we're working really hard toward that. A couple of things that I want to highlight about what we're doing. One is that the Black Immuno Week is a first step. We're basically planning for the rest of the year. And one thing that we have put together is a Black in Immuno Hub. It is something that we're still working on, but the idea is to create a database of all Black immunologists that want to file to be in the database in a central place that is going to sit on our website, where everyone can look for Black immunologists by subdiscipline, by level of career, or any other keywords. And I think this is going to help a lot of us to be visible and help research institutions to find candidates for their faculty search, and also try to connect the community with all these amazing researchers. So Black in Immuno Hub is going to be this database, but it's also going to be a place where we're going to try to create some networking events. And those networking events are going to be workshops. It's going to be also job fairs. We're thinking of bringing on some industry and companies to come and connect with Black researchers, Black immunologists. So we're trying to create a network that is going to be able to connect everyone and to allow people to come together. So, and that is one thing that I wanted to say. The other thing is, beside the sponsorship, what we're expecting from research institutions, but also, I mean, individuals can contribute by sponsoring, but we're also targeting, and we hope that they're gonna join in the fight with us, scientific journals and publishers and companies. And so basically all these different type of institutions can contribute by giving money for developing Black Immuno Week and Black in Immuno Hub. But beyond that, what we've thought is that there is an opportunity there for all these different institutions, companies, and publishers to start to take concrete steps to increase diversity and increase the environment for Black researchers, and especially in the field of immunology. So we have created the partnership program. And this partnership program has a very simple 3 rules, or 3, I don't know how to say that, but let's say a 3-point framework. One is to basically commit to collect and publish statistics about diversity in their local environment. So if I'm Department of Microbiology and Immunology Department of Research in a university, I want to partner with Black in Immuno. So that's one of the things that I want to do, start to collect this information and commit to publish it with Black in Immuno. The second point of the framework is to basically increase the amount of DEI trainings that are available for the people in this research community. And same thing here, we want numbers. That's the only way that we can evaluate how things are evolving. So we want institutions to publish the numbers of people that have been through trainings and how they're planning to increase that. And the third point of the framework is to basically have a plan of action. It doesn't have to be something that we would come up with. Any institution have a set of issues that they want to work on, so they can make their own plan of action. But this plan of action, we wanted to contain the numbers or the target numbers that this institution want to reach in the next 3 years, in the next 5 years, and in the next 10 years in terms of diversity and the proportion of Black individuals that they want to have in their institution. So it's really something that we're pushing forward, and we hope that a lot of institutions are going to be excited to partner with us with that. We think it's a simple framework. We know it's a lot of work, and for now it's something that is lacking. But it's very important that this first step is set so we can actually follow up how these institutions are doing and work with them to help them to improve the diversity and the quality of the environment in these institutions.
Just to add to what Joel said, I think really one thing we're trying to push is for accountability, because we know that during this moment, the recent month, there's been a lot of emails being sent out, a lot of plan of action. But really what we're hoping with this framework, we're keeping institutions accountable. So by publishing those numbers, by publishing your plan of action, you're basically holding yourself to it. You're going to continue what you said you were actually intending to do. So we hope that will help more visibility for Black scientists, but also keep all the institutions accountable for the things that they want to do.
Yeah, and with that accountability, not only will we feature you on our website, so when people need to figure out what graduate school should I go to, Black and Immuno says this, but we're going to give them a cool little logo of Black and Immuno, like Black and Immuno partner, that they'll be allowed to display. And so as they renew each year, they get to keep that logo. So then people know, okay, I need to look for that logo so I can start going to these locations. So it's just going to be, like Joelle said, it's a first step, but it'll definitely help and just get the word out and help people finding a place where that they can identify and that are doing the work.
It's like
I'm really impressed with how big. I mean, this is, like Alexis said, like dream big, go big. And I think when we originally started planning this, I don't think we talked about the hub unless I'm forgotten about it. I don't think we did. So
it came together in the last week. I think it was in the back of our mind since the beginning, but we've started to formalize it in the last, let's say 2 weeks maybe. And it's already on the website. There is already a page for it, but it's like work in progress. But you can already click on it and see that it's going to come together, and there will be this database and this program for social networking and all.
I like the idea of a curated seal. It's kind of like a good housekeeping seal of approval. I don't know if any of you remember that, but when I was a kid, that they say this is a good product, you can feel safe using this. So I like that very much. Is there anything else before we wrap up and say goodbye?
I wanted to ask each one of you. So you have this really nice plan for journals and for institutions. Can you just say one concrete thing that you think each of our listeners could do to join the fight, or whatever it is you want to talk about? So for each one of you, what do you think it is that our listeners could do?
I can start, I guess. I can start. I think I will think about my own graduate program and the things that I've seen that I think are helpful and are giving me hope. I think is listening. I feel like there have been a lot of assumption of this is why I think your experience is, or I'm in my bubble. This is not real. This is not happening to you. And I think for allies or people that are not Black to like basically being better at listening and trying to develop empathy. I know it's hard when you're not in somebody's shoes or somebody's experience. It's really hard to imagine that. But I think it's definitely important to be in those shoes, to at least listen to the person or think about their stories and believe in it, like it is real. Even though you don't feel it, you don't understand it, or you don't experience it, it is real. And acknowledging that goes a very long way, because then it makes me feel valuable. It makes me feel heard. So I think my thing will be like definitely starting to listen and trying to understand people's experience.
Yeah, to double back on what Elaine just said, I feel that silence isn't the answer, like at all. But to me, that's you being silent on the topic. It feels that you're not creating the environment where I personally can feel comfortable to be in. So speaking up, calling out microaggressions and racism when you see them, just holding yourself and other people accountable as allies. It will help take some of that burden off of us, because we can't hold the people that are doing the things to us accountable all the time. It's like it's a never-ending cycle, and we'll end up being exhausted.
Like T cells, right? Yeah.
So I guess I would maybe say, kind of in the vein of each one bring one, more of just on the same vein of, speak up, speak out, do the work. But I also want to say, just don't forget what it was like when you were in that stage. I feel like sometimes in academia, especially, even if you say, I don't know how to do this, people still assume you know how to do it. And so I know that was probably that's been my biggest struggle with my PhD, is just all these acronyms, all these forms, all this XYZ. And I'm like, I have no clue what you're talking about. So just not only advocating, but being true mentors and bring somebody with you, bring somebody that doesn't look like you to the field, so that we can continue to diversify. And you can continue to diversify and learn and become a better, more complete human, for lack of a better term.
I would say that the one thing I think everyone can do, and I think everyone should do, is to develop some awareness and understanding, and become an ally and work toward becoming an ally. And allyship is kind of a vague term, right? I mean, it can be a lot of things, but basically for me, as a scientist, allyship is really close to do science. Basically, allyship it makes you put yourself in a place where you admit that you don't know, right? And as scientists, that's what we do, right? We don't know about what the ILCs are doing. That's why we're doing research, right? So allyship is the same thing. It's like putting you in a place where you admit that you don't know about the experience of a Black person. You don't know how this and that can affect this person. So I think it's something that we can do as scientists, right? Because we do it for science. So we can do it also for in our relationship with other individuals. And then another part of allyship is to engage in learning in a perpetual way, right? And that's also what we do as scientists, right? We are going to read papers for the rest of our lives, right? We're going to do science for the rest of our lives to learn, right? So we can do the same about learning what are the experience of other people, what we can do to help people in going through some of this experience, what systemic barriers are, how we can break them, and all these things. Basically, for me, allyship is like doing science. So I'm very hopeful that scientists can pick up on allyship more easily than maybe the general population, because for me, there is this really strong parallel between being a scientist and being an ally. So yeah, I'm pretty excited for that, and I'm pretty hopeful that scientists are going to pick it up, and I think I see a movement. So I'm very hopeful, and I want to see how this goes.
I would say just be intentional about what you're doing. So if you are the head of your lab and you look around your lab and they're a homogenous population, maybe look into hiring somebody from an institution where you know there's a greater population of minorities, such as the HBCU or someone from Puerto Rico, because it's pretty minority serving there as well. So just being intentional about everything you do, basically, and knowing how it could affect others and how it will impact your lab or people around you in the future.
Last little tidbit, utilizing your platforms and your powers to amplify other people's or underrepresented voices. So that's like ultimate goal. And that's our ultimate mission for Black and Immuno. So.
Wonderful.
Yeah, and we thank you as well for using your platform right now to amplify our voices and our movement. And we hope that this is going to help to have more people knowing what we're planning to do and join us in the fight.
Yeah, absolutely.
Well, months ago, someone wrote to Twiv and said, "Why aren't you saying anything?" And that made me realize that we had to, right? Because sometimes it has to be pointed out, right? And the key is not that it has to be pointed out, but that you do something. And so I recognized that we had to do something, and that's why we have gone through the various Black in Immuno, and we have Black in Microbiology, Black in Virology, and we have tried to increase participants on all the shows as well. I think that your call to do something is what we're trying to do. We have a platform, and we are obligated to use it. So we're going to do it. No question. It's not just for this week either. It's forever.
So what's next with our relationship? I think you proposed that we can come back multiple times, and we're very excited. We're definitely gonna come back. That's for sure. But also we hope that other Black immunologists could join in and join your group. So I guess in the future, for people listening, if you're a Black immunologist and you want to be featured in this podcast, you're very welcome. And I think we're going to work toward having a Black immunologists to be part of Immune podcast in the future.
Well, we certainly love talking about papers, and when we have experts there to decode some of the stuff that we're not expert in, it's just wonderful. At least for me, I can speak to that. I really enjoy that.
Yeah, definitely. I think moving forward, that's what we'll be doing. We'll be having, any of the five of you, if you want to come back, you can pick a paper, we can talk about it. But then there's a lot of, PIs, really excellent Black scientists, Black immunologists that we can feature their careers and their trainees as well. Yeah. So, well, I think, this has been really great experience. I'm really thankful for you all for coming on and telling your stories. I'll kind of leave it up to Vincent to wrap up the end of the podcast.
You bet. This was a special episode of Immune. And you can find all the show notes and links to everything we talked about at microbe.tv/immune. If you have questions or comments, immune@microbe.tv. I want to announce a new Instagram account for all our podcasts, microbe.tv. That's the handle on Instagram. It's being curated by Lauren, who's a scientist in Philadelphia, actually. And she's making summaries of every podcast that we publish. And so she pulls up cool art and makes summaries and then throws out questions for people to engage. So I'm really grateful to Lauren for doing that. And once again, on Instagram, microbe.tv. And of course, if you like what we do, consider supporting us, microbe.tv/contribute. Our guests today from the University of Texas, MD Anderson Cancer Center, Alexis Mobley. Thanks for joining us, Alexis.
Thank you for having me again. It was so much fun, and I'm excited to be back. Talk about those ILC2s because they're the best.
From the University of California, San Francisco, Joel Babdur. Thank you, Joel.
Thank you so much. I had a great time, and yeah, we'll be back definitely.
From Icahn School of Medicine at Mount Sinai, Justine Knoll. Thank you so much.
Thank you for having us on this platform, and I'm excited to talk about science anytime in the future.
And from the University of Pennsylvania in Philadelphia, Yvonne Jean. Thank you so much.
Thanks for having us on. I'm really excited for Black and Immuno Week.
From the University of Chicago, Elaine Kouame. Thank you so much.
Yeah, thank you for having me. It was really fun. And like I said, I'll definitely be excited to come back and discuss virology and immunology.
Cindy Leifer's at Cornell University. On Twitter, Cindy Leifer. Thanks, Cindy.
Thank you. I really, really enjoyed this episode. I learned so much.
Stephanie Langle's at Duke University. Stephanie Langle on Twitter. Thanks, Steph.
Yep, thanks. It's been fun.
And Brienne Barker's at Drew University. BioProf Barker on Twitter.
Thanks. It was really great to be here, and I learned a lot today.
I'm Vincent Racaniello. You can find me at virology.blog. The music on Immune is by Steve Neal. Thanks for listening to Immune, the podcast that's infectious. We'll be back next month.